Hello, my name is Ellie Shaver from Montreat College, and today I want to share with you my research, designing a field test to determine chytrid fungus infection for amphibian populations in Loreto, Peru. First of all, what is chytrid fungus? Chytrid fungus is a type of fungus in phylum Chytridiomycota, which is a phylum in which fungi grow in soil and water, are microscopic, and they reproduce via zoospores. The specific species we're discussing today is Batrachochytrium dendropotidis, BD for short, which infects specifically amphibians. BD causes chytridiomycosis, a deadly skin disease in amphibians that has decimated populations of amphibians all across the world. It has been documented since the 1990s, and since then we have found it affects over 700 species, and it may cause up hundreds and hundreds of extinctions if nothing is done about it. It causes the greatest harm and is the most dangerous at higher elevations and cooler temperatures. Recent studies have shown BD is present at lower elevations and higher temperatures, specifically in the lowland Peruvian Amazon, than ever found before. Only a few surveys have been done there for the fungus since initial discovery in the Amazon, and it is hard to collect samples to test in a lab from such remote areas. Hence the need for an easy, low equipment field test. So thus, my project has a twofold objective. One, to survey for the chytrid fungus in the Peruvian lowlands in May 2024 at 2 sites along the Amazon River, and two, to find the best method for field detection of BD infections in amphibian surveys from 2 plausible testing methods. So in order to have the tests ready for Peru, we did preliminary testing in Buncombe County, North Carolina, over fall 2023 to spring 2024. We decided on 2 field tests, culturing and histological examination, and we tested these out on amphibians in the area, where we know chytrid is widespread. And our first test was the culturing test. We prepared agar plates using tryptone, agarose, filtered water, and tetracycline antibiotic in order to possibly grow chytrid fungus on. Then we went out and collected swabs from different specimens out in the field, brought those swabs back and inoculated the plates with those swabs, and then we let those sealed plates sit for a few days and see if the fungus grew on each of them. Our second procedure was the histology test. Each specimen that we collected had one small toe clipped, and we placed it in a tube to be brought back to our makeshift lab on the boat. Then we would take the toe out and, using a dissecting scope on a slide, to be placed on a slide to be looked at under a microscope. We looked at it under the microscope on every magnification and to observe to see if there were any spores present in the skin layer, which is a common symptom of chytrid fungus. Those were taken, we sterilized each specimen and discarded of it. We went out to collect samples on nightly surveys at our 2 field stations during the month of May, namely Madre Selva Biological Station and Santa Cruz Field Station. Specimens were collected by gloved assistants, bagged, and brought to camp to be processed quickly. Once we processed them, we, as soon as possible, returned each specimen to the location approximately where we found them. A new pair of gloves was used for each specimen in order to prevent cross-contamination of the fungus, which can be transferred by hand from one specimen to another. Each sample was labeled by location and number of sample collected, and we also collected the weight, the snout vent length, and the species of each specimen. For site-specific results, from Madre Selva we collected from 21 total specimens of 13 different species. We got toe clippings from the first 12, and we got 21 swabs from all of them. We found that 8 tested positive completely, which means In this chart, if they test positive, the specimen tested positive for both the culturing test and the histology test. 9 tested negative, which means neither test tested positive. And then we got 3 partial positives, which means that for this particular specimen, one test, maybe your histology test, showed a zoospore, so it tested positive, but nothing grew on the plate, and so therefore one test was positive and one test was negative, thus it's a partial positive, showing confliction in the test. At Santa Cruz, we only had partial positives and mostly negatives because none of our plates grew anything, which the other good but also suspicious. The histology showed that there was some chytrid fungus present. So samples were collected from 20 specimens at Santa Cruz, and no species tested fully positive. We had 2 partial positives where the histology test indicated presence of zoospores for 2 specimens, but the rest did not have anything on their plates or anything on their slides and were deemed negative. So 19 out of 20 were deemed negative. Plates at all, as I said a moment before, we had no growth on our plates at all at Santa Cruz. For Madre Selva, by day 3, we looked at the percent growth across the plate on each agar plate that we had to see how much matter was growing on the plate. Madre Selva had a variety, mostly very small amounts of growth, and some plates that were almost completely covered in growth. Our culturing testing finds, especially with our culturing test, lots of different colored growth on our plates, including purple, yellow, white, and a crazy vibrant red for one particular gladiator frog, which shows, unfortunately, that we have some contamination on our plates, because chytrid fungus is not going to grow all those different colors. It's mostly going to be the white, the simple white growth that you can see in the bottom right here. As mentioned just before, both tests need much more refinement and examination before they can be used officially to test for chytrid fungus infection in the field. Testing with a control group using a known chytrid fungus strain would be very effective to make sure we hone down on the tests and know exactly what we're looking for and how to diagnose from the tests. Use of a hemocytometer for measuring plate growth would be very important for the culturing test. Be able to see how many zoospores are swimming around in the plate matter. Histology test, we would want to have a repeatable method for preparing slides, such as using a portable microtome to cut the skin. Some other method for better toe slicing and correct staining methods to more clearly see what was inside the skin cells. Thank you for listening and getting a peek at all these cute little frog photos.